What's up, Internet? Dr. Dean Radin is an American parapsychologist. He's one of these guys who worked on clandestine government programs like Project Stargate. He was a researcher at Princeton, and now he is a parapsychology researcher for the Institute of Noetic Sciences, which was founded by former astronaut Edgar Mitchell. Also, for the rest of this video, I call him just Dean, which is no disrespect meant by I should call him Dr. Radin, but I was just being lazy. Anyway, Dr. Radin appeared on the Joe Rogan podcast recently, and he made some shocking claims, one of which being this.
So they were turning something off. We still don't know exactly what it is, but something about their makeup was turning off psychic sensitivity. And so one of the people on our project was a specialist in the genetics of societies. Different societies have different genetic makeup. And he found that, to our surprise there too, that countries that were exposed to Christianity, the longer they were exposed, the more this intron sequence was there, the mutation. We started thinking, well, how does that make any sense? And then suddenly we understood. The Inquisition had systematically looked for people over hundreds of years who had these abilities, and then they killed them.
Okay, so I took a look at the actual paper that he is referring to here. Yeah, so what this paper actually, they started with like 3,000 candidates. They narrowed it down to 23 people that 13 of which they thought were actually, were like pretty sure they were psychic, after interviewing them. They're pretty sure they're not like con artists. And then 10 people, who like never have had a experience or something. Anyway, point is they identified like this one interesting mutation, and the mutation is in the intron adjacent to an exon in the TNRC 18 region on chromosome 7. This variation and alteration of GG to GA, so that would be okay, actually I just looked up GG to GA. What does that abbreviation mean? And what that means is it's a change from homozygous to heterozygous. Something like a one like base pair change, I think. So yeah, I'm not so interested in all the survey methodology and stuff like that, because what the really interesting thing is like the gene, like what is this mutation you actually identified? Okay, so the variant of interest identified in this pilot study is located in the intronic region adjacent to the TNRC18 gene. Due to its proximity, it might be involved in regulating that gene's expression. TNRC18 is highly expressed throughout the body, including the brain.
Gene and ontology annotations indicate that the gene product has multiple functions, including chromatin binding and transcription, regulatory region sequence, specific DNA binding. This variant is quite common in the population sampled by...
Blah, blah, blah. So that's interesting. Okay, so yeah, a mutation from homozygous wild type to a heterozygous, meaning like the wild type should have both base pairs the same, but the okay, yeah, so GG, so that's guan. Okay, now I get it. It's guanine. Guanine, is that how you? Let me. Guanine. Guanine and guanine. Yeah. Guanine and then adenine. Right. So it went from guanine to guanine adenine mutation. So it's a one base pair mutation. The wild type should just be all, both guanine. So you go to an adenine, and like that's an interesting mutation to identify. That could, cause something. The study is basically a correspondence with a very small sample size that people with this adenine, where they should have a guanine, don't seem to experience any psychic phenomenon or abilities. That's the claim. So the claim TNRC 18, that there's like right next to this gene that functions, there's this mutation, and we maybe see something as a result. So Dean, Hal Puthoff, probably even could say like Salvatore Pais, or some of these guys that are like in the Joe Rogan type scientist category. I don't knock what they I don't they're good at presenting things in a way that's going to be very appealing to the general public and to like funding sources. And I don't want to be a hater on that, right? Like it's a part of the game, and it doesn't mean that your science isn't valid necessarily, right? But like it really makes me think of, or reminds me of, this news story from a while back. Quantum yin yang shows two photons being entangled in real time. So what a lot of the public didn't understand about this is like this this pattern that they're showing here was something that they fabricated. They made the yin yang design so that the whatever the experimental results would be real visible. But the general public like just kind of thought like, oh, this is proving like that the yin yang thing is like fundamental, showing it like as a fundamental shape in the universe, right? That's not what this actually did, so yeah, Dean, I think in terms of what he actually has here, and in terms of like how the public might watch Joe Rogan and like interpret this is there's definitely some gap in there, right? Or let's rewind a minute. So, what the reason this appealed is I've been getting lately so I've been getting into epigenetics. This is really just this has a lot to do with anti-aging research that's recent. I don't think the connection, when I was studying biology at USC, the connection was not as well understood, I don't think. But basically, a lot of this aging is believed to be that your cells lose differentiation because the markers that tell your body how to, like, what part of the DNA to read and transcribe, manifest, basically, changes and gets out of whack, and that is bad. So, what these genes like T and RC18 do, they can encode histones, like proteins within a histone, and the histone is like what the DNA is wrapped around, and so the parts that are like tightly wrapped around these histones are not used, basically, whereas the parts that are like open, just like a open long segment of DNA, are then transcribed. Like the transcription factors bind much more easily, and you see a lot more transcription, like based on where they, like the histones, like what they wind up versus what they leave like straight, kind of determines like what genes are being expressed. So anything in the DNA that like encodes the manufacture of these proteins that are then going to wind the DNA around, is, going to be significant and have significant effects. So I went a little deeper into this, and I looked in the Human Genome Database to see what type of activity I could find around this sequence. Anyway, all this gold activity happening right here totally implies that this mutation does something in the brain. So basically this one mutation, GG to GA, so a guanine to an adenine, is gonna affect probably this H3K27AC histone, so I would love to go deeper into this, but yeah, let's take a look at what Claude says about this. I used AI a little bit in working on this, as one does, and there's like a segue there, or like a connection, because AI is a hype man as well, right? Like AI will tell you anything you do, it tells you like, this is amazing, right? You're making an amazing discovery, doing amazing work right now. So that's, you always got to dig through that. And as I dug through this with Claude, did that process, really dissected and got to the bottom of what Claude was actually telling me. Two things really stood out. One is what we saw in this chart. This in this, all in the center here. It's the histone H3K27AC. Anyway, H3K27AC is a promoter. It is going to actively mark an area to be transcribed. Okay, so the other interesting thing about TNRC18, so in terms of published literature, TNRC18 is actually identified with a completely different histone, H3K9ME3. And that was in this paper. TNRC18 engages H3K9ME3 to mediate silencing of endogenous retrotransposons. So that's really interesting. So what that means is this paper is saying that this gene controls or engages a histone that's a silencer. So, and what it's silencing is retroviruses, endogenous retroviruses. So these are viruses that have incorporated into our DNA and become part of us. It's like somebody had an infection in their sperm or egg cell, some ancestor a long time ago, or an alien genetically engineered you, but the only one of those is currently scientifically accepted. So somebody had a virus and this incorporated into the DNA, and then the body learns to silence this part of the DNA, or not, right? So what's really interesting is that so many of these endogenous retroviruses are expressed in the brain. We're kind of running, it's one of the, when we talk about the alien thing, it's really kind of weird that we would want these viruses, this virus DNA active inside our brains for normal function, unless it was some type of genetic engineering, right? It shouldn't add anything, but yet it makes some sense with the evolutionary process also. So somebody got this virus, and then their offspring was smarter, and then they passed on their genes, and you could see that, but it's weird. Most of the time it should be, you would think it would be a negative. So yeah, this is all, the BAH molecule of TNRC18 specifically reads H3K9ME. So these are, so part of this paper, TNRC18 mutation causes death in mice. So the same type of mutation that we're looking at, that Dean Radin's group has identified. So not the same mutation, but the same type. They tried in a mouse embryo, and it killed them. They couldn't survive. Anyway, yeah. So we have evidence, or like a little bit of data suggesting both that this gene can be a suppressor, particularly suppressor of viral DNA that's incorporated into our own genome, and that it can be a promoter, of another histone that's going to mark DNA to say, like, yes, express this DNA. So yeah, seems like an important gene, like any, and again, this is like where the science is like the hype versus the reality, because like, I mean, if you look at populations, you find any gene mutation, if it does anything, that's gonna be a significant finding. And then if you find a mutation on like an important gene, like, oh, this gene does all this stuff, well, it's probably gonna be worth looking at. Like, so the evidence about this psychic bit is like maybe like an unnecessary but interesting addition. I mean, from a pure like research standpoint. But yeah, I am into this gene and this mutation, and I would love to be able to answer the question, which I still have not been able to answer. Does this mutation do anything or not? And yeah, Dean Radin and his group, I think that their response would be, give us more money, and we would love to answer that question. But, hope maybe some of us can answer it. Maybe I can answer that question, and somebody will donate money to my YouTube channel instead. So for the next session, I think the next suggestion of AI was to query Ensemble's variant effect predictor to see like what are the effects of this variant. So I am probably going to save that for the next video that I do on this, but I would like to go as hard as I can on being able to say, does this mutation do anything? What histone, if any, does it actually affect? And how is like the histone formed from the mutation different than the wild type, and like what difference does that make in the brain? So yeah, please support my channel. I may even try to put like a donation link if people want to donate to science. But also, I'm going to put a link to the socials for Continental Breakfast. That's my short film that I did that we premiered in October at a film festival. We won Best Art Comedy at the Austin Short Film Festival. And then I had to buy a duplex, and I got completely, just financially just wasn't in the great spot to be doing all the submissions, like to more. So we're back doing submissions, and those guys just look at some. I'm about to say something bad. Many of the wonderful film festival people watch your movie and really care about how good your movie is, but also some of them look at the social media and just see how many followers you have. So if anybody watching this video could please follow us on social media, that would be helpful. Or donate money to science. Thanks, y'all. See you next time.